Immune complex-mediated hypersensitivity, type 3 hypersensitivity reactions. A type 3 hypersensitivity reaction is caused by immune complexes, which consist of antibodies bound to antigens. Antigens and the resulting immune complexes float freely in the blood. As a result, these immune complexes are more precisely termed soluble immune complexes. The antigens involved are either external, representing foreign antigens, or internal, also termed self-antigens. Matching IgM, or usually IgG antibodies, bind to the antigens and form complexes of various sizes. Generally, these immune complexes are effectively removed from the blood, especially by macrophages, which engulf and digest them. However, smaller immune complexes are more likely to remain in the blood circulation. The circulating immune complexes tend to deposit on the blood vessel wall, especially in small vessels. Once immune complexes are formed, they rapidly initiate the complement system. This family of plasma proteins acts as a cascade that mediates further activation of immune cells. Subsequently, a series of reactions are triggered, with the most decisive involving neutrophils for type 3 hypersensitivity reactions. Neutrophils are attracted by activated chemotactic complement proteins. Using the Fc receptors on their surface, neutrophils bind to the Fc portion of antibodies in the immune complex. This binding leads to the release of destructive substances that cause cell damage in the vicinity of the deposited immune complex. Damage to endothelial cells as well as to the basement membrane of the blood vessel wall increases its permeability, resulting in leakage into the surrounding tissue. This provokes further inflammation and subsequent tissue damage. Two distinct features help to discriminate a type 3 hypersensitivity reaction from a type 2 hypersensitivity reaction. First, the components initiating the type 3 hypersensitivity reaction are initially soluble before depositing on the surface of the vessel wall. Therefore, tissues with small blood vessels and thin vessel walls are most affected by type 3 hypersensitivity reactions. Examples include the kidney, which filters blood, as well as joints, which filter plasma to produce synovial fluid. In contrast, the antibodies in type 2 hypersensitivity reactions are directed against cellular or matrix structures and therefore have fixed targets. Second, activation of the complement system is more rapid and much stronger in a type 3 hypersensitivity reaction. Serum levels of complement proteins can therefore be used to monitor the course of the type 3 hypersensitivity reaction.